Hello, everybody. Am I audible? Can someone let me know if I'm audible? Yeah, okay. We'll start the session. Yeah, I hope my screen is also visible. If there is any issue, or if I'm not audible, or if the screen is not visible, please turn on the mic and let me know. Because once I go into presentation mode, I won't be seeing the chat box very often. Okay. So please feel free to turn on your mic and speak whenever there is an issue, or if you want to ask something or have any doubt, also you can turn on your mic and talk. Okay, so let's begin. So this is week 6, experimental biotechnology from NPTEL by Professor Vishal Dwivedi, IIT Guwahati, and I'm one of your TAs, Sumati Raviraj from University of Hyderabad. So this is my email ID, as you know. Any queries, any doubts, you can also mail me to this. So this week, the syllabus was affinity chromatography. So let's just go in brief, just have a recap of what affinity chromatography is. So affinity chromatography is a powerful and highly specific separation technique. Just a second. So it is basically used to purify biomolecules, and it is based on the ligand and uh, target specific interactions. So it is based on the biological recognition, that is the specific and reversible interaction. So it is very important that the reaction or interaction, whatever is there between the target and the ligand, should be reversible reaction, because whatever sample you have, whatever protein or any biomolecule that you want to purify, it has to bind to the uh, partner, binding partner, but also at certain condition it has to be unbound from it so that you can purify it. So it has to be reversible interaction between a biomolecule of interest, that is your target, and a complementary ligand to it, which will be the binding partner. So this is immobilized on a stationary phase. The stationary phase could be different types of resins or beads. So agarose beads, sapphirose beads, saphacryl beads, many different variety of beads are available based on the type of sample, based on the type of target, tags, binding, and type of purification that you require, and the downstream you have to use your sample for. You can choose the material of interest and go ahead with the purification. So this technique, the affinity chromatography, is also called as a lock and key chromatography, because like this is also a very common example or an analogy that is given usually even for your enzyme substrate binding. It's a lock and key model. It's similar to that. So the every key has its own lock, and every lock has the specific key. So you cannot open a random lock with a random key. It has to be their binding or their target pairs. So in the same way, your target of interest will only bind to its binding partner. So that is the reason you can then easily purify only your specific target of interest. So this is because the target molecule fits specifically with the ligand, just like how a key fits perfectly with the lock to open it. So that is why the lock and key analogy is also given for the affinity chromatography. Then coming to the basic principle behind it, the principle is specificity affinity interaction, and it is between two biological molecules like the target and the ligand. So the ligand here is immobilized on the column, on your beads or on your resin, and your target molecule is present in your sample mixture. When the sample mixture passes through the column, so this is your column, your sample is passed through the column which has these beads of your ligand. So the target then specifically binds to the immobilized ligand, and then other molecules that do not recognize the ligand wash through, and then the bound target is later eluted by changing the buffer conditions or pH or ionic strength, or even by adding a free ligand. So, like I said, it has to be a reversible binding so that you can later, once bound and all the unnecessary things are washed out of the column, later you have to finally regain your protein. So, you have to elute it. So when it's reversible, it will be unbound from your ligand. Maybe if you change your buffer, or maybe if you change your pH of the buffer, or any kind of excess salt addition, or by adding even a different ligand, which could be more specific to your target protein of interest, which can then unbound it from your column, and then you can elute it out. So, therefore, the separation is achieved due to mutual recognition forces, which could be either hydrogen bonds, van der Waals forces, or ionic bonds or hydrophobic interactions, etc., depending on the type of target we are handling. So this is a simple illustration. This is your sample molecule, sample mixture, which has your protein of interest. You can see the tiny dark blue or green uh, bits here, which is your protein of interest. So it you can see it's with a different mixture of all other biomolecules in it. So now you have to separate only your specific those rectangular types of protein. So the sample is then flowed through the affinity column in the binding buffer. So then you have your ligand, which is specific to your protein of interest, which binds to it. So the other molecules do not bind to your ligand molecule. So therefore, uh, the unnecessary excess biomolecules or non-specific biomolecules are washed out. So the sample is then washed through the column to remove unbound components. Then the target molecules bind to the affinity ligands that is already present in the column. Then once you wash it, your unbound components are removed. Then now you have only your target that is bound to your ligand. So now you have to make it come out of the column. You have to make it lose its interactions with the ligand. So then you use your elution buffer, which may help in breaking the bonds between your ligand and the target, so that you can isolate your or purify your protein of interest. So your elution buffer is applied to disrupt the interactions between the affinity ligand and the target molecule. So now you can see the interactions have been broken, so your target molecules are eluted out. So the purified target molecule is eluted from the column. So this is the basic principle uh, on how an affinity chromatography works and how you can use it to purify your target or your biomolecule of interest. So, affinity chromatography is a highly selective separation technique based on specific biological interactions between two molecules, usually termed as ligand and target molecule. So, your target can be anything, it can be a protein, it can be a receptor, it can be an enzyme, antibody, it can be polysaccharide, anything. So, any biomolecule. So, it was first introduced. Just a second, I lost the pointer. Yeah. So it was first introduced in the 1960s and is widely used in protein purification, enzyme isolation, and in various other biotechnology aspects. So often, It is considered to be the gold standard for purification because it is highly specific. Because, like you said, every lock has its own key, so every target protein will make use of a highly specific ligand. So it is very specific, and it will give you very highly purified protein or target which is free of any other sort of contamination. So it is considered as a gold standard for purification when a high degree of specificity is required. So the mechanism, like we discussed, is immobilization. A ligand is which is specific to the target is covalently bound to a solid support, which can be anything, like we discussed your stationary phase, which is your beads, your agarose beads, sapphirose beads, or acrylamide beads. So you can see here your bead is attached to the ligand, which will be specific to your target. So this beads are usually of like common materials that are used for making beads, which is agarose, sapphirose, and saffacryl, and then they are tagged with your uh, ligand of interest. So that is the reason you can use specific ligands to any specific biomolecule of interest, because all these beads can be attached to different types of ligands. So yeah, so that is your immobilized or your stationary phase, which has your beads attached to your ligand. And then binding takes place where the sample mixture is applied. Then the target molecule specifically binds to the immobilized ligand on your bead. Then you wash it away to remove impurities and unbound elements, and then you elute to get your target molecule out. So the target molecule is then released by disrupting the interaction between your target and your ligand. And the elution can be of different types, like we discussed. We can change different conditions like pH or ionic concentrations, or you can add a free ligand or a substrate which will help in replacing it. It could be more uh, it could have more affinity to your ligand or substrate than the already bound one. So it could be a competitive replacement, and then your protein of interest will elute out. Then you can also change the conditions, like you can alter the pH, you can change the ionic strength of the buffer, or you can, you can play around with the polarity of the groups, and this will help weaken the binding. Then you need to regenerate your column. So once you have eluted out your protein of interest, you have to equilibrate your column again so that you can reuse it for the next time. So these are the basic mechanisms: immobilization, binding, washing, elution, and regeneration. So to go step by step, first you have column preparation, where the ligand is immobilized on the support matrix. That is, your ligand is your specific for your target, and your support matrix is your beads or your resin. So first you add your ligand to your support matrix, and it is bound to it, immobilized on it, and then you load your sample. So sample loading is done. So your sample mixture is applied to the column, then binding takes place, the target molecules bind to the immobilized ligand, and then you do your washing so that the non-specific and weakly bound molecules are washed out. Then you do your elution to bound molecules which will be now specifically released, and then regenerate your column so that the column is restored and it is in the same condition as it was before, like that is free of any molecules or any particles bound to it, and it is in now in a proper optimal condition for next use or for storage. So there are several applications of your affinity chromatography. We can do protein purification to get pure forms of proteins like antibodies, enzymes, hormones, etc. And then we can use it for enzyme studies where we want to isolate the enzymes with specific substrates or cofactors, and then receptor ligand studies can be done. We can characterize the interactions between the proteins, and then purification of monoclonal antibodies, of course, using protein AG affinity columns, and then nucleic acid purification. So, like I said, not only proteins, we can also separate different biomolecules like nucleic acids. So, to use that, you need to use your complementary DNA RNA strands, or you have some things like aptamers, which are complementary to your specific sequences in your DNA or RNA. So, this can also be done, and then also has certain applications in diagnostics. So it can be used in biosensors and immunoassays and biomarker detection assays, etc. So these were the applications, and now coming to the limitations of it. It's quite expensive because you need the column itself is pretty expensive, and then you need to have your ligand, specific ligands, and the ligand immobilization and column preparation is the most expensive part of it. Then stability issues. So if you do not know how to run the column, how to store the column, how to regenerate the column, it can be difficult to maintain it, and the column might lose its stability, and then you cannot reuse it properly. So these columns can be ranging anywhere between thousands to lakhs also. So when you have such expensive columns, and if you're unable to maintain it properly, it will be such a waste of resources. So you have to be very careful while using these columns, and make sure that you don't dry it, you don't, leave it in leave it in inappropriate buffers, use proper storage conditions, etc. So stability issues are there. So immobilized ligands may lose the activity after repeated use. So the ligand that you've added, sometimes after a certain time, they might become inactive. So then you can't again use it. So there is like a particular number of uses for which it will be helpful, and then depending on the ligand that you have chosen, uh, it its reuse capacity will be determined. Then limited binding capacity. So overloading reduces the efficiency. So when you run your column, obviously, like the activity or anything, when you're continuously using it, and or if there is like uh, there are a lot of mm, different molecules in your mixture or in your sample, if it's too much for the column to handle, then the efficiency might reduce. Then non-specific interactions. So sometimes you don't know, depending upon the lysate you have, or depending upon the source of your sample mixture, uh, you might have some unknown interactions that might be happening. If you don't know all the components in your sample, then sometimes you might not know the non-targeted interactions. So at that in such cases, the specificity decreases, so you won't get like the complete pure form of your target. So sometimes non-target molecules bind weakly, lowering the purity. Then illusion challenges. So illusion, you are obviously changing the condition so that you reverse the binding. So sometimes harsh conditions like extreme pH, high salt, etc., may denature the sensitive proteins. So these conditions are necessary for your protein to be eluted out. But sometimes, if your protein is extremely sensitive, change in such kind of conditions might affect the protein's activity or the protein's structure or form. So, in case downstream you're planning to do some other experiments with the protein, if you want to do like an activity assay, or if you want to characterize the protein structures, then you have to be extremely careful that which conditions can be altered to that specific protein. so that you don't lose the protein's characteristics or you don't denature or degrade the protein by changing such conditions. Next is, although it's a highly specific technique, it's not universal and it requires prior knowledge and specific ligand for the target molecule. So, like I said, if you don't know what sample it is, if it's like completely new sample out of the blue, something you want to find out in that whether your particular target of interest is there or not. But then, if you don't know what you want to elute, or if you don't have the complete information of your target and its binding capacity to your ligand, and if you don't know what other components are present in your sample which might have or may not have interaction with the ligand you have chosen, so that might become an issue. So you cannot always use this for every type of sample, only for the samples with for which you have all the information about its specificity, interacting partners, off target effects. If you know all this, then this can be applied. But for like completely new samples or new completely unknown targets, you cannot make use of this technique. So, in summary, affinity chromatography is definitely a powerful and highly selective purification method, and it uses the principle of ligand receptor interaction or recognition for the purification, and it gives high purity in a single step. But it is expensive and requires prior knowledge of specific interactions. So, this was a brief intro and steps and a basic principle behind affinity chromatography. So, now coming to the assignment questions, let's discuss regarding those topics. So, the chromatography technique works on the principle of mutual recognition forces between a ligand and a receptor. So, this is only the affinity chromatography that we were discussing. So, this technique relies on specific and reversible biological interactions, which is also often called as mutual recognition forces. So, this is an interaction between a molecule of interest and a complementary binding partner. So, these binding partners can be anything until it is specific. Like, for example, you can use the interactions of an antigen and an antibody because you know antigen and antibody are binding partners and they are very specific. Antibodies of which are raised against a specific antigen only bind against that antigen. So in the same way, enzyme substrate complexes, like I also told you, like the lock and key when we were discussing, for the enzyme substrate interaction, also we use the lock and key model as an analogy. So you can also use the enzyme substrate interactions or enzyme cofactors or enzyme inhibitors as interaction partners. Then we definitely have the receptor and the ligand, or receptor and its substrate. Then we have lectin and sugar moiety. So lectin are like these uh, molecules like which are present in the a few of the samples from maybe like plant extracts or bacterial or peptidoglycan or anything like that. So lectins are molecules which have interactions with the sugar moiety groups. So we can make use of that if you have any kind of poly saccharide kind of targets that you have to purify. Then applications, highly selective purification for proteins, enzymes, antibodies, nucleic acids, or hormones. And the phrase mutual recognition forces between a ligand and a receptor directly describes the basis of the affinity chromatography. So, this is nothing but affinity chromatography. So, this is a simpler uh, table which compares different types of chromatography. Your affinity chromatography, the basis of separation is specific ligand receptor or biomolecular interactions. And ion exchange, the other options which were given is your ion exchange, which is basically reliant on the electrostatic charge difference, and hydrophobic interaction chromatography, that is your HIC, is based on the hydrophobic patches or hydrophobic nature of your sample. And the gel filtration chromatography is basically your exclusion chromatography on the basis of size and shape. So the one which has mutual recognition ligand receptor interaction is your affinity chromatography. Next is the receptor molecule which binds, which has the affinity towards biotin containing enzyme. So this is avidin. So biotin and avidin, we have your uh, one of the binding pairs. So avidin is a glycoprotein from egg white, and it has extraordinarily high affinity for biotin, which is vitamin B7. So avidin and biotin are usually used in many techniques, not only in affinity chromatography, even in several other antibody antigen, uh, other interactions studies, or like binding partner studies. So avidin and biotin are very common partners that are used in different experimental setups. So the dissociation constant for this is 10 power minus 15 molar. So this is a very strong non-covalent interaction. And this property is exploited in biotin avidin affinity chromatography as well, where labeling and detection assays can also make use of this kind of interactions. Then other options that were given were 5 prime AMP, heparin, and con concanavalin A or con A. So 5 prime AMP, so this mimics the adenine nucleotide part of coenzymes. So it is used in affinity chromatography for nucleotide binding proteins like kinases, dehydrogenases, etc., and not for biotin. And then heparin, it binds with nucleic acids, growth factors, lipoproteins, etc., and not specific to biotin. Then con A, it is a lectin. So this binds specifically to sugar moieties like the alpha D mannose and alpha D glucose residues. So it is used for either your polysaccharides or your glycoprotein purification, and not in case of biotin binding. So the applications for this, like I said, since they have very strong interactions, biotin avidin is used in several detection assays and biochemical assays. So few of them are in case of protein purification of biotinylated enzymes, antibodies, and nucleic acids. And it is also used in case of pull down assays to capture the protein complexes. Then, excuse me, they are also used as labels in different other detection uh, and identification visualization purposes. So biotin can be conjugated to antibodies, DNA probes, peptides, etc. Then avidin or streptavidin conjugated to enzymes or fluorophores are used, so that when the avidin binds to the uh, biotin which is present on the antibodies or the probes, it will give you some sort of detection, like when tagged with fluorophores or chromophores, etc. So your avidin or streptavidin is conjugated to enzymes or fluorophores. Example, streptavidin HRP, streptavidin FITC. So this is used for detection in case of ELISA, Western blot, immunohistochemistry, etc. So these biotin avidin are very famous, popular pair, which is used in many techniques like your Western immunoblotting, your ELISA, your even your in your IHCs, in your immunohistochemistries, your DAB staining, or immunofluorescence, etc. So this is very useful in many of the techniques. Then also immobilization, where biotinylated molecules can be immobilized on avidin coated surfaces for biosensors, microarrays, and diagnostics assay. Then drug delivery and nanotechnology. So even in drug delivery, the avidin biotin bridges are used to attach your ligands, your targeting ligands, and imaging probes or drugs to nanoparticles, which will then be used which are used as delivery systems. Then there are also certain limitations for this, although it is very widely used. Sometimes, when used in case of your affinity, harsh elution is needed because the interaction, like we spoke, is 10 power minus 15 molar interaction. The dissociation constant is, so it's very, very, very strong interaction. So you need like really harsh changes in the pH or any solution buffer conditions so that you can break the bond. So this, like we discussed, may sometimes become problematic to your protein of interest, your target of interest, if they are sensitive proteins. So avidin is glycosylated and also positively charged. So this can also cause non-specific binding sometimes. So streptavidin is often preferred from Streptomyces avidini, and it has no carbohydrate moiety, so it reduces the non-specificity due to positive charges, and it still has high specific affinity to biotin, just like avidin molecule. Then the cost of these reagents are relatively high. This is definitely there in case of any type of affinity chromatography techniques. This was about biotin and avidin and their applications. Coming to the next topic, it is a ligand L and a receptor R forms ligand receptor complex RL. The expression, yeah. So the expression to calculate the dissociation constant would be dissociation constant is nothing but concentration of your receptor into concentration of your ligand divided by the concentration of the receptor ligand complex. So how this formula is derived is your ligand and your receptor are binding partners. So your ligand receptor binding reaction will always be in equilibrium state similar to your enzyme substrate reaction. So you have your receptor, you have your ligand, and they will be in stoichiometric equilibrium with the receptor ligand complex. Your here, when we are speaking just the receptor and ligand, it means unbound forms. So your receptor here is your free receptor, which is not bound to your ligand, and your ligand here is your free ligand, not bound to your receptor. And when we are talking about RL, it is the bound forms of both your receptor and your ligand. So your R is free receptor concentration, L is free ligand concentration, and RL is ligand receptor complex concentration. Then the dissociation constant, the definition of dissociation constant is nothing but uh, it is when the equilibrium constant for the breakdown of your complex. So basically, it tells you how much affinity the specific ligand and the receptor has. So the dissociation constant is the equilibrium constant for the breakdown or the dissociation of a complex. So it goes both ways. So it can also be RL, your complex, when it dissociates into free ligand and free receptor forms. So what is the equilibrium constant there? So by definition, the dissociation constant is concentration of your receptor and concentration of your ligand divided by the concentration of your receptor ligand complex. So here we have, in the numerator, we have concentrations of the free species, that is the dissociation products. And in the denominator, we have the complex, which is the associated form. So it is basically the concentration of your dissociated forms divided by the associated form. It's a ratio between dissociated and associated forms in simple terms. So it is nothing but RL divided by concentration of the RL complex. So, how do you interpret it? Like, how do you know when you have a dissociation constant? How can you understand the affinity from it? So, the smaller the KD, that if you have the numerical value of your KD is small, that means the complex is highly stable. It has more affinity. So it has high affinity between the ligand and the receptor. Therefore, it is highly stable. The complex is more stable and the KD value is more small. When the KD value is large, that means the affinity between them is very weak and the complex is unstable and the binding is also very weak. So the units that it is expressed is molar, m. So it is a numerical and a molar. So that is why in the previous case, your biotin, sorry, yeah, that is why you see your dissociation constant here was 10 power minus 15. So it is such a small value. It's like 0.0000014 times 0 and then 1. So it's 0.000001. So that is such a minute value. So you can imagine how much is the binding capacity, how much strong affinity they have. So the more smaller your KD value, that much more the strength of the binding partners is. Then we also have something called as occupancy equation. This is just for information. So this is important when you have like drug studies or if you have to know your binding capacity of your drug. So total receptor concentration is nothing but R total is equal to your free receptor plus your receptor that is bound with a ligand. Makes sense. Then the fraction of the receptors bound is theta. That is the occupancy. That means when you have certain number of receptors, how many receptors are able to bind to the ligand? That is the fraction of the receptors bound. So that is given by theta. So that is concentration of your receptor ligand complex divided by the concentration of total receptors present. So using the equilibrium expression, we can derive from this theta is equal to concentration of your free ligand divided by your dissociation constant plus the concentration of your free ligand. So if you know the information regarding your ligand, you can get the basic understanding of how much receptor binding has taken place. So here at L, your KD, so if your theta is half, so half of the receptors are occupied. So theta is half, which is equal to 0.5. So half of your entire receptor population that is present in the sample or in the solution are being occupied. So this is why KD is often described as the ligand concentration at which 50% of the receptors are bound. So you can see here in the final derived equation, we represent it in terms of the concentration of the ligand. That is why when you uh, describe KD, you generally describe it as half the concentration of the ligands, where uh, it is the concentration of the ligands where 50% of the receptors are bound. So this is basically about the dissociation constant. and how it can be interpreted either in this way or this way. Then there are various applications for this, like I said, in terms of drug studies, in terms of biochemistry, protein ligand binding studies, and drug receptor affinity determination, and interaction in enzyme kinetics, antibody antigen interactions, etc. So, do you guys have any questions till now or any doubts? No? Okay. Okay, if you guys have any doubts, you can just raise your hand or just turn on your mic and let me know. So, moving on, the next is the chromatographic technique, which gives maximum yield to purify the specific target protein from the cell lysate. So, from the cell lysate, you have to purify your target protein with maximum yield. So, obviously, when it is specific and you want your pure maximum yield of your pure form, you will go for affinity chromatography, which yields the highest and purest portion or fraction of your specific target protein. Since it is highly selective receptor ligand interaction, antigen antibody enzyme substrate biotin avidin, etc., and even more complex mixtures from cell lysate. So, like I said, it always depends on the efficiency, efficacy, everything depends upon the type of sample that you have. So, when you have a cell lysate, your cell lysate has everything in your cell. It has your proteins, it has your peptides, it has your free amino acids, it might have your, truncated versions, it will have your sugars, it will have your nucleic acids, it will have other uh, small organular complexes, it might have your receptors, your substrates, everything, all sorts of biomolecules is present in your cell lysate. So, even from a complex mixture like your cell lysate, the target protein can be isolated in a single purification step with very high specificity. And other options like your HIC gel filtration, ion exchange are not very specific and or may not yield high purity because your HIC is basically dependent on the hydrophobicity. So it separates protein based on the surface hydrophobicity and it does not specifically purify one particular protein. So it is useful as an in intermediary step, maybe, but not highly specific to one target. Then your gel filtration chromatography is basically a size and shape selective, so it's not highly selective and cannot isolate one protein from a mixture of lysate. And the yield also will not be in pure form. Then your ion exchange is based on the net charge. So, just like your hydrophobicity, it will basically separate out everything with similar charge. So, it will not be able to specifically pick that one protein of interest. So, high capacity and useful for bulk purification, but the specificity is less when compared to affinity methods. Next is in dye affinity chromatography column, blue saffrose matrix is coupled to Cibacron blue. So this is a dye. So blue saffrose is a dye ligand affinity chromatography matrix, and the saffrose or the agarose beads are covalently coupled to Cibacron blue F3GA synthetic triazine dye. So why is the background blue? So it is because the structure of the background blue mimics the structure of nucleotide cofactors like NAD plus, NADP plus, FAD, ATP, etc. So, therefore, it binds to many nucleotide dependent enzymes like dehydrogenases, kinases, albumin, etc. etc. So, this makes blue saffrose widely used for purification of albumin and nucleotide binding proteins. Other than this, other options like bromophenol blue is not a binding partner in your affinity. It is a tracking dye which is used in electrophoresis, and your Kumasi blue is also uh, uh, a kind of visualization dye which is used in Bradford, or it is used to stain your SDS page because it binds to the proteins. And trypan blue is a dye which is used to check cell viability. It is not specific for any kind of purification technique. Then Cibacron blue, a few other details of this is it is a synthetic anthraquinone dye, and it is bright blue in color, and it is reactive triazine dye family. It belongs to triazine dye family. And then the chemical structure, like I said, it is similar to your cofactors. So it has a ring, it has a triazine ring, and it has sulfonate groups, which makes it water soluble and reactive as well. Then it forms covalent bonds with hydroxyl groups of agarose, and giving us a blue sapphirose for affinity chromatography. Then other properties of blue, Cibacron blue, is strong but reversible binding to nucleotide binding proteins. Non-specific binding to some other proteins might be there, so washing and elution conditions are very important. So such kind of non-specific binding can be eliminated by increasing the washes. And then inexpensive, it is also inexpensive when compared to other affinity columns and uh, resins and beads that are used, and it can be an alternative to true biological ligands. Then uh, main applications are protein purification, especially in cases of albumin kinase dehydrogenase, and removal of albumin from serum. Since albumin binds strongly to Zibacron blue, it can be used to remove it from the serum. And affinity chromatography, when natural ligands are too expensive or unstable, this can be used as an alternative for that. Then also there are a couple of limitations. Non-specific binding. Not all interactions are due to nucleotide mimicry. Sometimes some proteins stick through hydrophobic and electrostatic interactions because this is also reactive. And sometimes the selectivity is low when compared to true ligand based affinity chromatography like antibody, antigen, etc. Then next is cyanogen bromide. So the coupling agent that is used for coupling polysaccharide matrix in bioaffinity chromatography is cyanogen bromide. In bioaffinity chromatography, a ligand such as an antibody or an enzyme substrate or inhibitor, is covalently attached to an inert support matrix, which is basically your saccharose and polysaccharides or agarose and saccharyl, etc. So, this is to achieve the polysaccharide needs to get activated so that it can form covalent bonds with the ligand. So, then we have cyanogen bromide, which is the most common coupling and activating agent which is used, and it reacts with the hydroxyl groups on the agarose and saccharose beads or the matrix, and then it introduces reactive cyanate esters or imidocarbonates. So these groups then react with primary amine groups on the ligand, forming the stable covalent linkage. So this is why cyanogen bromide is used. Then other options like aluminum bromide, hydrogen bromide, and potassium bromide. These are all uh, acids and other salts which are not used for affinity chromatography. So aluminum bromide is a Lewis acid, so it is used in organic synthesis and not during coupling. And hydrogen bromide is a very strong acid and has no role in affinity matrix activation. Then potassium bromide is a neutral salt. So it is used sometimes in IR spectroscopy for sample preparation and not as a coupling agent. So coupling agent that we use is cyanogen bromide. Does anyone have any doubt? Someone's mic was on. Does someone have any doubt? Yes. Okay. Yeah, so we were talking about cyanogen bromide. So this is a coupling agent that is used for polysaccharide matrix for activation of the matrix. Next is DTT. So DTT is dithiotretol. This is used to break the sulfide bridges, disulfide bridges. So it disrupts the antibody structures due to breakage of disulfide bonds. So DTT is a strong reducing agent and it reduces the disulfide bonds between cysteine residues in proteins. Antibodies are stabilized by interchain and intrachain disulfide bonds. If you know, remember the antibody structure, it is like a Y, and then you have the light chains and the heavy chains. These are bridged by disulfide bonds. So between the heavy chains we have disulfide bonds, between the heavy light chains we have disulfide bonds, then within the domains of heavy and light chains also we have disulfide bonds. So breaking these disulfide bonds with DTT disrupts antibody quaternary structure and it then separates the heavy chains from the light chains. So others like hydrogen bonds, they do not like are they are not affected by DTT. So hydrogen bonds stabilize protein secondary structure, but they are not affected by DTT. Salt bridges are ionic, so they are electrostatic and are disrupted by changes in ionic strength and by pH, and not by DTT. Van der Waals forces are hydrophobic interactions, so they are also not reduced by any kind of reducing agents. So one other agent which we use generally is BME, that is beta mercaptoethanol, which is also reducing agents. So this is a small comparison between DTT and BME, and when you use DTT and when you can use BME. So DTT and BME are both used for the same purpose, that is, they both are reducing agents. They are used to break the disulfide bonds and to denature the proteins. So these are used also in SDS page in sample preparation in the lamellae buffer, sample buffers to break the disulfide bonds to denature the proteins into individual subunits, like how in case of antibody we got separated heavy and light chains. In the same way, peptides which are bound by disulfide bonds will be now broken and we will have a more linear form of your protein, which is the denatured form. So DDT is preferred when you are working with sensitive proteins and you need a stronger agent, and it is more precise control during the reducing environment, and it is also less smelly than BME. BME has, if you have worked with it, you know it has a very pungent odor and it's like really smelly and it spreads around very easily in the atmosphere. So DTT is less smellier than that. BME is used when you want to do something in large scale, if you because DTT is very expensive, so you can go with BME when you want a huge amount to be used, and it is inexpensive and reduction. So it also does it, but it is relatively less stronger than DTT. So this is a small comparison table. So the thiol groups, DDT has two thiol groups and BME has one thiol group. And reducing effect is strong and efficient in case of DTT, and it's weak. And because of that, you need more amount of BME when you have to reduce the disulfide bonds. Then the concentration is 1 to 10 millimolar in case of DTT because it's very strong, and because we need excess of BME since it's a weak reducing agent, we have to use at least 50 to 100 millimolar. Then the byproduct of DTT is stable cyclic disulfide. The byproduct of BME is, however, free disulfide dimers. And volatility and smell. DDT has a mild odor and it is less volatile. BME is very volatile and is very pungent and very smelly. DTT is commonly used in SDS page and sensitive assays. BME is used in routine reductions and also in SDS page. Then we have something called as a booster dose and how it enhances the adaptive immune response. So booster dose injection repeat is nothing but repeated exposure to the same antigen. This helps enhance the adaptive immune response. In rabbits or mammals in general, B lymphocytes undergo something called as clonal expansion, and then they have class switching and affinity maturation after repeated exposure to the same antigen. So this leads to more plasma cells and B cells, and resulting in stronger, faster, and more specific immune responses upon boosters. So why not others? Why not monocytes? Why not NK cells? Why not T lymphocytes? Because monocytes, first of all, are part of innate immune system and are not significantly enhanced by boosters. NK cells also innate system and they do not have clonal expansion. And T lymphocytes do get activated, but however, the hallmark of booster vaccination is to produce more antibodies. So B cells are the ones which are responsible for production of antibodies. So strong B cell response and high antibody titers are seen when you have booster doses. I think I need a charger. So, excuse me, I'll just get my charger. Yeah, hope I'm still connected. Yeah. So, do you guys have any doubts till now? Okay, so let's continue then. Hope I'm audible. Okay, so just a quick recap about primary and secondary immune responses. So, the primary response is the first time when your body is exposed to antigens. So you have your naive B cells, that is your B lymphocytes, which encounter the antigen for the first time. So initially, the antibodies that are produced are mainly IgM antibodies which have low affinity. So it takes several days to weeks to mount a measurable amount of antibody response. The titer is very less, and very few memory B and T cells are generated. However, when you have a booster dose or a booster injection, you have a secondary response or a booster response. This is because of the repeated exposure. You have the repeated secondary response. So when you have this, your memory B cells, which were already formed during the primary response, will now quickly expand because they had already encountered this. So they have the memory of that exposure. So now they don't need as much as time that they needed for the first time because it was new and need more time for them to identify, clonally expand and everything. But now, since it was already exposed, now they expand rapidly, and the response is much faster and stronger. And this time, predominantly IgG antibodies are produced. So, this have high affinity due to the affinity maturation. Then antibody titers quickly rise and reach higher levels than primary response. So, what happens to B lymphocytes after booster dose? They undergo clonal expansion, that is, memory B cells divide rapidly upon re exposure to antigens, and then affinity maturation happens, that is, somatic hypermutation in the immunoglobulin genes produces high affinity antibodies, and then class switching occurs from IgM to IgG, IgA, or IgE depending on the type of signal that is stimulated. And with IgG being more prominent in the booster doses, it has higher affinity, and plasma cell differentiation occurs. More plasma cells are produced, and massive antibody secretion occurs, and long-lived memory B cells persist in the circulation, providing lifelong immunity in most of the cases. So your antibody profile after your booster injections is basically, in primary you have IgM, which is slow and has lower titer. Secondary you have rapid rise, high titer, IgG predominant, and long lasting antibodies with long lasting memory and greater specificity and affinity. This is why booster injections are standard in case of polyclonal antibody production in rabbits to generate high affinity IgG antibodies in large quantities. So, practical example in case of rabbits is rabbits are immunized with an antigen, and after 2 to 3 weeks, that is the time in which your primary response develops, then we have booster injections given to them so that now the secondary response is generated, and the rabbits B lymphocytes produce much higher amounts of high affinity IgG antibodies. Then the blood is collected, serum will be rich in polyclonal antibodies right now, which can be purified and then it can be used in research and diagnostics, etc. So, after repeat booster injections, B lymphocytes undergo clonal expansion, affinity maturation, class switching, differentiation into plasma memory B cells, making them the most developed cell type. So this results in a rapid, high titer, high affinity IgG antibody response, which is a hallmark of a booster immunization. This was about booster. Then next is the chemical compound used for activation of polysaccharide matrix is epichlorohydrin. So this process is used to create reactive cross-linked polymers for application such as affinity chromatography. So we have polysaccharide matrices like the agarose beads, which are often used as supports. So to covalently attach a ligand, the matrix must be first activated. So epichlorohydrin is commonly used for this. It reacts with the hydroxyl groups of polysaccharides. It forms reactive epoxide on the matrix surface. So these epoxide groups can then react with the amines, thiols, and hydroxyls of the ligand, which will help the ligand bind on the matrix. So this creates a stable covalent linkage. Other options like hydrin, anhydrin, halohydrin. So hydrogen is not used as a activation or a coupling agent in chromatography. Ninhydrin is a reagent for detecting amino acids, which gives a purple color and not for matrix activation. And then halohydrin is one which has this functional group, but it is not a standard reagent for matrix activation. Epichlorohydrin itself contains a halohydrin group which cyclizes to epoxide in reactions, but only halohydrin is not a coupling reagent. Then regarding the titer, antibody titer in case of indirect ELISA. So antibody titer is nothing but the concentration or relative amount of antibodies in a serum sample. So the most widely used method to determine antibody titer is indirect ELISA, that is enzyme-linked immunosorbent assay. So in indirect ELISA, what happens is antigen is coated on a microtiter plate and then serum sample is added to it, which has the antibodies. So the secondary antibody, so this will be your primary, and secondary antibody will be an enzyme labeled IgG antibody against IgG antibody, and this binds to the primary antibody. So the substrate is added and the color development is proportional to the antibody bound on it. So by performing serial dilutions of the sample, highest dilution that gives a positive signal is the antibody titer. That means the more you dilute a sample, still if it's able to bind to the antibody, that means the sample is that rich in antibody. The sample has so much antibody that even after several hundreds of dilutions, you can still see the antibody antigen interaction in them. So the principle of indirect ELISA is antigen is immobilized on the microtiter plate and serum which contains the antibodies, primary antibodies are added to bind to the antigen, and then the enzyme conjugated secondary antibody binds to the primary antibody, and then addition of chromogenic substrate produces color which is detected by the optical density and is proportional to the amount of primary antibody present, which is again proportional to the amount of antigen present. So generally these are the steps. You can go through it if you're interested. We'll just skip it for now. So how do you determine the antibody titer? So you have something called as endpoint titer. So this is the reciprocal of the highest serum dilution that gives an OD above a predefined cutoff. So you have a positive signal which is your predefined cutoff. Till that OD you can have your diluted sample. So your cutoff is commonly set by this formula, that is it is the mean negative control plus 2 or 3 into XD negative control. So if you have to have more stringency you can go with 3, or if you are not that stringent you can go with 2. So for example, if you have diluted your sample 1 is to 6,400 times, and this is the dilution where you have gotten your last OD value before your cutoff. So your OD is greater than your cutoff, and this was the dilution 1 is to 6,400. So your titer value would be 6,400. Alternatively, you can also have titer as EC50, which is the midpoint. So this is from a 4 parameter logistic fit to the OD versus log. So no need to get into much details of this. It is useful when comparing samples quantitatively. You can also have a standard graph like protein estimation. So if you have a reference antibody of a known titer value or a known concentration, then you can compare that with your unknown and find out the titer of your unknown. Then in all these cases, you need to have controls. So you need to have negative control serum, that is pre-immune serum or a serum from a naive animal which was not exposed to any kind of antigens, so it will not have any antibodies, so that it doesn't give any false positive results. So then you have blank wells where you have no serum to check if your reagents are giving any kind of background. This is all regular steps which we do. Then you have a positive control in which you know that you have your antibody, a known antibody present in it. And then secondary only control. So here you do not add your primary, you add directly only your secondary. So this shows if your secondary antibody has any kind of non-specific binding. So in that case, you might get a higher signal than it should actually be due to non-specific binding of your secondary. So that is the reason to avoid this, we have something called as blocking. So just like in case of Western blot or immunofluorescence, etc., where you block the regions where there might be non-specific binding of your secondary. Similarly, here also you have a blocking. So this is also another control which you can use. And of course, whenever you do ELISAs or these kind of quantitative tests, you always do it in duplicates or triplicates so that any sort of handling error or measuring errors are eliminated. Then you would have your data handling. So you have your plate layout, you have your run dilutions and duplicates, including the control. So after all the background deletion normalization, you will get a particular OD value before your cutoff, and that particular dilution will be your titer value. So that is it about this. Now coming to Cepheros bead. So Cepheros bead, this is a common matrix that is used in affinity chromatography, and it includes the antigen coupling during antibody purification. So in antibody purification, the most widely used support is your Cepheros, that is cross-linked agarose beads. So protein A Cepheros and protease G Cepheros columns are gold standard in immunoglobulin purification because protein A and G ligands are covalently coupled to Cepheros beads, which are then bound to the FC region of the IgG antibodies. So Cepheros beads are chemically stable, inert, and hydrophilic and allow easy functionalization. So with cyanobromide or epoxy activation, you can activate these columns. Uh, then also why specifically Cepheros is because it is purified agarose polymer and it has a porous bead nature. It contains hydroxyl groups which can be chemically activated using cyanogen bromide or epichlorohydrin to couple antigens or ligands on it. It is hydrophilic, inert, porous, stable. So it minimizes non-specific protein binding, does not react with proteins unless chemically activated, allows antibodies even 150 kilodaltons to diffuse in and bind inside bead matrix, withstands pH and buffer changes, which is important for purification. So these are some advantages that it is commercially available in pre-activated forms in cyanogen bromide Cepheros, NHS Cepheros, epoxy Cepheros, etc. It can be packed into columns easily with good flow rates and large-scale purification. It also has high binding capacity, allows purification from mg to even grams in case of antibodies. So this is the coupling we already discussed, and then other options like cellulose beads, sucrose beads, galactose beads. These are used in some chromatographies, but the yield or the mechanic strength is very less and has less common applications for antibody. And sucrose is not used for coupling. Sucrose is used mostly in density gradient centrifugation. And galactose beads are used in lectin affinity, not in antibody affinity. So this is generally for sugars, that is like galactose binding proteins, etc. So these are few applications of Cepheros beads and antibody. So purification of IgG subclass, antigen antibody specific, support matrix in immunology and biochemistry is commonly Cepheros 4b and 6b. Then few limitations, it's expensive when compared to other crude supports, but specific. And beads may compress under high pressure, and it is not ideal if you want to further go for HPLC. And then antibody may sometimes denature in illusions conditions if it is too harsh. So this is usually in everything. So just to summarize, the bead is most commonly used in antigen coupling in antibody purification, and it is because of its hydrophilicity, stability, porous nature, and ease of chemical activation. It is superior to others like cellulose, sucrose, and galactose in terms of binding capacity, mechanical strength, and versatility. Then coming to most appropriate method for peptide sequencing is, okay, so this is we generally do mass spec for peptide sequencing, but other older techniques like Edmond degradation are still used for short and pure peptides. So mass spectroscopy is most predominantly used right now due to its speed, sensitivity, and ability to handle complex mixtures, and others like crystallography, NMR, IR, high HPLC currently are not. So crystallography is basically to determine the three-dimensional atomistic structure of the molecule, and it is not for sequencing. NMR is also similar to crystallography, and it is used to analyze the structure and dynamics of the biomolecules in solution, and it is not used to determine primary amino acid sequence. It can, however, provide some information to identify short range of sequence tags. So short sequence tags can be NMR can be useful. IR spectroscopy is, however, for the functional groups that are present on a molecule based on their vibrational modes. It can be used to study the secondary structure of the protein, but it cannot determine the amino acid sequence. HPLC is a separation technique used to purify the isolated peptides and proteins based on their physical and chemical properties. It is a critical step in preparing sample for sequencing for mass spec, but it is not by itself a sequencing method. So HPLC can be coupled with Edmonds degradation, and or HPLC can be coupled with mass spec to uh, do sequencing. So peptide sequencing involves determining the order of amino acids in protein or peptides. So the most classical and widely used method is Edmond degradation, where the N-terminal amino acid is labeled and cleaved without disrupting the peptide bonds. Then the released amino acid derivative is identified, and HPLC is then used to separate and identify each amino acid derivative with high resolution and sensitivity. For very short peptides, NMR can sometimes distinguish between the residues using their unique chemical shifts, but it is mostly for their 2D, 3D, 4D, and isotopically labeled samples one can map such residues. This gives partial sequence connectivity, but not a straightforward readout like Edmonds degradation or mass spec. So HPLC with Edmond degradation or mass spectrometry are used for sequencing. NMR is mostly for structural dynamics and not for complete sequencing. Then next is immobilized metal affinity chromatography. So this works on the principle specific to electron donating groups of proteins, usually like histidine side chains and transition metals. So these are immobilized on a resin, and this is often done using a chelating agent like nickel, NTA, or IDA. So the common metals used are nickel, cobalt, zinc, copper, and iron. So nickel is most common, high affinity for polyhistidine tags. Cobalt is more selective than nickel, less non-specific binding. Zinc sometimes is used for protein requiring milder elution, and copper has strong binding but can cause protein oxidation. Then iron is sometimes used in phosphoprotein affinity, but however chromium is not used for metal affinity. So chromium does not form a stable coordination complex with histidines, so this is not suitable for metal affinity chromatography. They are less biocompatible and can be toxic or even oxidizing. Thus, chromium is not used for column charging in iMac. So this is a table comparing the strength, selectivity, advantages, drawbacks, and applications of different metals in case of iMac. You can just go through this. Next is bioaffinity chromatography. So here Purification occurs through the specific binding between a ligand and a target. So, first we have loading, then we have washing, and then we have elution. So, when loading, target molecule binds to the immobilized ligand, other proteins does not interact pass through. So then we need washing. Washing removes unbound or weakly bound proteins. Only the specific ligand receptor complex remains in the matrix. Hence, fractions collected in this step contain only the unbound protein molecules. Then, in illusion, the bound protein is released. So, diluted fractions contain pure receptor ligand protein of interest. So, flow through or wash has unwanted unbound proteins, and illusion has purified target proteins. You can see here mixture, and then when you load your sample, then you have your binding of your specific ligand protein, and whatever is unbound goes in the flow through and in the wash. Then during elution, you have purified protein which was bound to your ligand specifically. Then coming to adjuvants. So adjuvants are substances mixed with antigens to enhance immune response. So by themselves, many purified antigens, especially proteins, peptides, or polysaccharides, are weak immunogens. So we need adjuvants to increase the immunogenic nature of the antigens. So prolonging antigen persistence at the injection site, slow release takes place. So adjuvants helps in enhancing antigen uptake by antigen presenting cells. It stimulates innate immune response, cytokine release, danger signals, promotes adaptive immunity leading to stronger antibody production or T cell response. Then some common adjuvants are France complete adjuvant, very common FCA, antigen plus mineral oil plus killed bacteria. Very strong immune booster dose. Then alum is also used. Alum is aluminum salts. So widely used in human vaccinations and safe and enhances humoral immunity. Then monophosphoryl lipid A, MPLA, is a detoxified endotoxin. So this derivative is used as an adjuvant. So, an antigen, when not enough on its own to raise an immune response, is connected to an adjuvant so that it gives a higher antibody titers, long lasting effects, and more robust immune response. So, I guess this is the end of all the questions. I hope we have covered all the questions in the assignment. Uh, does anybody have any doubt? Any doubts, anyone? Okay, if there are no more doubts, I think we can end the meeting. Thank you for joining, everybody.